everyone. Today we are going to review the foundations of the MRX/MRN complex. We will be dissecting the ideas of our paper titled Complex Formation and Functional Versatility of MRE11 of Budding Yeast in Recombination. The MRX complex, which is the complex discussed in our article, is a protein complex composed of the proteins MRE11, RAD50, and XRS2. XRS2 is the yeast homologue of the MBS1 protein that is found in humans, meaning the complex is called the MRN complex in humans. These three proteins are important for the formation and processing of double-strand breaks, aka DSBs, which are damages in DNA that are introduced prior to meiotic recombination and are lethal to the cell if not repaired timely. The MRN complex plays an important role in DSB repair, which promotes cell survival. Prior research indicates the binding of RAD50 and MRE11 to carry out these processes, but further research is necessary to define the full MRN complex form and conclude that XRS2 is a complex component. As previously mentioned, double-strand breaks are damages in DNA that contribute to cell mutations and can be lethal to the cell. DSBs are introduced during meiosis to initiate recombination. The MRN complex is necessary for meiotic recombination and works alongside the SPO11 protein, which catalyzes DSB formation. DSB processing includes cutting out DNA nucleotides to create substrates for binding factors that are needed for repair processes such as homologous recombination and non-homologous end joining. The MRN complex plays a vital role in sensing DSBs and contributes to activating protein kinases that participate in the DNA damage response.
In this experiment, the researchers formulated two hypotheses. The first one is that MRE11 forms two different complexes with RAD50 and XRS2, one that forms DSBs and one that processes DSBs. The second one is that MRE11 displays multiple functions alone and when interacting with different proteins. The key experimental techniques include immunoprecipitation, Western blotting, and gel shift assays. Immunoprecipitation uses antigen-antibody interactions to isolate the protein of interest out of solution. Western blotting analyzes if the immunoprecipitated protein is the protein of interest. Lastly, gel shift assays, or EMSA, can detect protein complexes through protein-DNA interactions.
Complex formation between MRE11, RAD50, and XRS2 was studied using Western blot analysis of immunoprecipitates from burying cell lines. In Figure 1a, an anti-MRE11 probe was used to pull protein out of solution for each cell line, and the precipitates were ran on 3 gels, with each gel being stained specifically for either the MRE11, XRS2, or RAD50 protein, as can be seen labeled below each gel. The cell lines being tested were wild-type cell lines in meiosis and mitosis and deletion mutants for each protein, indicated by the delta symbol. In the first gel, we can see that an MRE11 band is not present for the MRE11 deletion mutant, but is present in all other cell lines. This result tells us that MRE11 does not need either of the other two proteins in order to be stable in solution. In the center gel, we can see that the bands specific for XRS2 are not present in the MRE11 deletion mutant or the XRS2 deletion mutant. This result tells us that MRE11 must be present in solution in order for XRS2 to be stable in solution, but RAD50 is not required for XRS2 stability. In the last gel, we can see that the band specific for RAD50 is not present in the MRE11 deletion mutant or the RAD50 deletion mutant. This result tells us that MRE11 is necessary for RAD50 to be stable in solution, but XRS2 is not necessary for RAD50 stability. In Figure 1b, this experiment was repeated using an anti-XRS2 probe to pull proteins out of solution. The first gel shows that XRS2 was unable to be isolated from the XRS2 deletion mutant, but was able to be isolated in all other cell lines and wild type. The second gel shows that MRE11 is unable to be isolated from the XRS2 deletion mutant or the MRE11 deletion mutant, but is able to be isolated from the RAD50 deletion mutant. This result tells us that XRS2 can bind to MRE in the absence of RAD50. Lastly, the third gel shows that RAD50 was unable to be isolated from all 3 deletion mutants. This result tells us that the XRS2 protein can only interact with RAD50 in the presence of MRE11. Combining the results of these gels, it was concluded that MRE11, XRS2, and RAD50 bind to form a complex, with MRE11 acting as the centerpiece of the complex.
So in answering what parts of the MRE11 sequence bind to DNA, an EMSA or gel shift assay was utilized to figure out if the protein bound to either M13 single-stranded DNA or PUC19 double-stranded DNA. And so the graphic here is an agarose gel, which displays DNA, and in this gel, DNAs which bind proteins travel slower. And so the MRE11 sequence was split into 3 parts, S1, S2, and S3. They were subcloned and fused to a GST or gluthionine S transferase gene. And so in this experiment, GST acts as kind of a negative control. And so you can see S2 and S3 binding both single-stranded and double-stranded DNA because it's higher up on the gel. And they were named, respectively, DNA binding site A for S2 and DNA binding site B for S3. And you can also tell that S1 did not bind any protein in this case by looking at the DNA markers present under the GST lanes in lanes 1 and 5.
Endonuclease activity was measured by adding MRE11 mutant proteins with circular viral single-stranded DNA. It was converted to linear form, incubated at various times, and analyzed using a gel electrophoresis. We can see that the wild-type and 11.5 mutant, which has a deletion from the C-terminus, resulted in extensive degradation of the substrate, as there is a singular band for linear DNA only. These results show that the C-terminal region of MRE11 can be deleted for endonuclease activity. Mutant 1158, which has a mutation in the phosphoesterase consensus amino acid sequence, and mutant 11.6, which has an 11 amino acid deletion in S2, did not digest the single-stranded DNA. The mutant 1158 results indicate that the phosphoesterase motif needs to be intact for the activity. However, MRE11.6 results showed no ability in this assay, although it has the intact phosphoesterase consensus region. The second figure utilized a gel shift assay to measure single-strand DNA endonuclease activity of the MRE11 segments previously described. The 11.5 mutant, which has a deletion in S3, can be referred to as S12, which confirms there is nuclease activity as there is a band for linear DNA. We can see that there's no nuclease activity in S2, 3, or other segments alone. Thus, the main takeaway is that S1 and S2 are both needed for endonuclease activity. To demonstrate MRE11 exonuclease activities, MRE11 was incubated with 5' labeled ssDNA and measured over time. Figure B shows the excision of nucleotides from the 3' end, indicating the presence of 3' to 5' ssDNA exonuclease activity. However, we do not see the formation of the labeled mononucleotide on the gel. Therefore, MRE11 does not exhibit 5' to 3' ssDNA exonuclease activity. In Figure C, dsDNA exonuclease activities were measured in the MRE11 mutant proteins using DS oligodNA labeled at the 5' end of one DNA strand. The 3' end of the labeled strand was digested with MRE11 when the end of the strand was blunt or recessed, but it was not digested when the end overhung by 4 nucleotides. This demonstrates that 3' to 5' dsDNA exonuclease activity depends on the structure of the DNA ends. The MRE11 5' mutant and wild-type proteins similarly displayed 3' to 5' dsDNA exonuclease activity, but the MRE1158 and MRE11.6 mutants did not display dsDNA exonuclease activity for the same reasons mentioned in the previous slide.
The MMS sensitivities of mutant and wild-type MRE11 cells were examined to study MRE11 repair mechanisms. MMS is an alkylating agent that indirectly produces double-strand breaks, and MRE11 is known to play a role in repairing damage induced by MMS. The survival of the cells on yeast growth plates was measured at various MMS concentrations. As you can see on the graph, mutant 11.5, which has the ability to process double-strand breaks for repair, is resistant to MMS similar to that of the wild-type cells. This suggests a repair mechanism that requires a cell to have nuclease activity, which mutants 11.6, 11.58, and 11 delta do not have. On the other hand, the intermediate mutant 11.6 has partial resistance to MMS and cannot process DSB ends. This partial resistance suggests the likelihood of an MRE11 repair mechanism that does not require the protein to have nuclease activity. Further research is necessary to affirm this speculation.
The following major conclusions can be drawn based on a discussion of the MRN complex thus far. First, MRE11, RAD50, and XRS2 form the complex for double-strand breaks, with MRE11 acting as a binding core for processing. However, due to structural changes in the complex, MRE11 does not act as a binding core in formation. Second, MRE11 has 2 binding sites, Site A and Site B. Site A is for processing. We know this because MRE11-6 mutant, which has a mutation in DNA bind Site A, could no longer process. Therefore, we conclude Site A is necessary for processing. Site B is likely utilized in DSB formation because MRE11-6 does not have Site A, and MRE11-58, which has a Site B, was sufficient enough for DSB formation. Third, the S1 and S2 sites are necessary for endonuclease activity. The C-terminal region of MRE11 can be deleted for nuclease activity. However, the phosphoesterase motif needs to be intact for nuclease activity. And finally, the ability to process DSBs affected by MMS resistance and partial resistance of mutants defective in processing suggests a nucleus-independent repair mechanism. Moving to further research, one question that arose from our paper was what proteins bind to the S3 domain of MRE11 during meiosis. The scientists in our study were able to isolate 3 proteins that bound to S3 during meiosis, and they speculated that 2 of these proteins could possibly be proteins known as RAC102 and RAC104. The 2003 Zhao study listed on this slide confirmed that RAC102 and RAC104, along with SPL11, were proteins involved in a meiotic recombination complex. Another question from our paper was how does the MRN complex signal double-strand breaks to the cell? The 2012 Schiller study listed on the slide examined this question and speculated that a conformational change between MRE11 and NBS1 may signal to other molecules the presence of a double-strand break.